kind of back up a little bit and just look at the big picture. And so I'm going to draw like a table on the board, which, so if we look at a living cell, most of you have heard the stat that 70% is water. So that means the dry weight of the cell is in that 30% that's left over. And that includes 1% is metal, another 1% are the sugars, basically, but the little ones, mono and oligosaccharides. And then we have about 0.5% each for free amino acids that are not part of proteins or peptides, and free nucleic acids that are not part of DNA or RNA. And then we actually have we have like double that amount for free fatty acids. And then we get about 0.5% for just other small molecules. And the rest, the other big 25% that we haven't covered, I know that doesn't totally add up just right, but this is all just approximations. The other 25% is going to be macromolecules. Some of which are polymers, but some of which are just really big structures, like such as lipids. So half of the cellular dry weight is protein. And that is what Rosetta was originally designed to handle. Is like let's model proteins and design proteins, but there's still a lot left in there. So in humans, we have about 1% RNA, but that number goes up to about 6% in bacteria. So that's kind of interesting. And then for DNA, it's about 1%, and then 2 to 5% is lipids. And I was going to say there's more in mammals here. And here is more in bacteria. Okay, and then 2% is all the big sugars, the polysaccharides. And that number goes up to 33% in plants. But for bacteria and mammals, this is roughly how the percentages look. So there's a lot more that we have to deal with than just the protein. And this number is also a little bit deceptive because we say this is 15 to 18% is protein, but the fact of the matter is about 70% of proteins in mammals anyhow have sugars attached to them. So they're not really just straight protein, they're not just amino acids. They're like a polypeptide with branches of sugars coming off, or post-translational modifications to those amino acids. So things are a lot more complicated than just to branch them up into these groups. So if you want to have a full picture of biological function, you have to understand the actual structures that you're dealing with, and the fact of the matter is the actual structures are not just peptide, they're not pure lipid, they're like glycolipids and glycoproteins in most cases, or there's post-translational modifications and such things like that. In addition, we mentioned over here we have these small molecules and we have metals, and these are both really, really key. Metals, I would say, do two main things. One would be they act as strong positive charges. So they're good for binding things like ATP. So e.g. binding of ATP. So that's something that you're not going to generally get from just your amino acids. The amino acids, we have 20 canonical amino acids. There's a lot more chemistry you might need to do in a cell to stay alive than just what you have with the 20 amino acids. So if you throw in metals, you get some extra advantages. Another thing the metals are involved in are electron transfer. So if you're doing oxidations or reductions, or if you're doing photosynthesis, metals are highly important to these sort of processes. And they're also important in some cases for the structure of the proteins. So if you want to do a de novo fold of a protein that has a metal involved, it might be really important to explicitly model that metal in the metal binding site to get the proper folding. Or if you're doing the docking of ATP to a protein, it's really important that you have that metal in there. So I don't know if you've heard these terms before, but the terms they use, if you have a protein that is kind of complete and whole, it's called a holoprotein. And that includes cofactors, like the metals, and then the apo is missing it. And so a lot of times when people model things, they unfortunately model the apoproteins. They might be missing really important information that is in the holoprotein. So I mentioned metals briefly, and in addition to metals, we also have other small molecules, such as vitamins and cofactors. And I mean, I don't know how much organic chemistry or biochemistry all of you have had, but just some quick examples. If you want to do reductions, you need to have a source of hydride. And so we use NADH for that. You've probably seen NADH or NADPH. Those molecules are really key to the chemistry that happens in the protein, and they bind in a very particular place in these proteins. And if you ignore them while you're doing design, you might be missing the really key interactions that are required to figure out what you're trying to design. So anyway, I just think it's really important to kind of consider that there's a lot more out there than just peptide. So we're going to be doing a workshop a little bit later that will answer the question, well, what do you do if you have one of these cofactors or metals, if you want to load that structure into Rosetta, how can you do that? And we're going to play around a little bit with how to load ligands in. And of course, I just said ligand. In addition to cofactors, things that actually do chemistry, and metals that are kind of part of the structure of the protein, you also just have the substrates of your enzymes. And a lot of the work in Baker Lab has revolved around putting a substrate molecule into the enzyme and then designing the enzyme around that substrate. So you need to be able to know how to load in that molecule to do the design. And I know you've learned about design, but loading the ligand is a key part of that in many cases.
We did some of the enzyme design papers last week.
Great. Okay. So you know what I'm talking about, right? So we've covered a little bit the importance of small molecules and metals, and just a broad overview and why we would care about that. But what about all these other macromolecules? So besides protein, what if you have this giant strand of RNA or DNA? Or what if you have a giant chunk of cellulose that you would want to model? That means, in that case, you have a polymer. And Rosetta, as you know, is residue-based. So we've kind of broken down the peptide into small little chunks that we can handle. There's no point having to load in all this different information if all the alanines share the same atoms. You can just kind of store that in the same kind of data structure in the computer. So we would do the same thing for polysaccharides. If cellulose is made of a bunch of glucose, we only need to have one glucose residue type, and then all of the residue, all the glucose residues in that polymer can share that same information. And so that's kind of the method that we go about using in Rosetta to kind of manage the memory and handle it. So we can do that with all these cases. RNA and DNA, of course, only have 4 or 5 residues. They're pretty simple. Polysaccharides will get really crazy, and we'll look at that in a second. So what I'm going to do is I'm going to drop on the board some of the kinds of polymers that people have loaded into Rosetta, and just see if you can recognize sort of what would have to change, what would stay the same, what kind of methods we can use to handle them. So for one, well, I'm going to draw this. I'll just kind of start out with an amino acid here. All right, so this is an amino acid residue. These are supposed to be squiggles to go from the lower connection to upper connection. Usually everything we've done has to do with L amino acids. Well, there actually are D-amino acids in nature that can happen in certain situations, generally not in proteins, but you could synthesize them if you want to do some sort of fancy synthetic stuff. And they do exist in some small molecules and some antibiotics. So if you had a D-amino acid, how do you think that would change how things would work in Rosetta? If you just loaded this in and you scored the protein, what sort of changes would you expect to see, or what problems would you run into? So the only difference is the stereochemistry right here has changed, right?
You don't want that extra oxygen.
Oh, right, technically. Yeah. So, some of we have the repulsion, attractive energies. Those probably shouldn't matter, right? Because those are based on the atoms. But a lot of some of the scoring methods in the Rosetta score function are based on information from the PDB database, right? Like the Dumbrack rotamer libraries are a good example. And so those look at your phi and your psi angles, and then they look at the rotamer that you have for that side chain, and based on these Ramachandran plots and statistics, you get a score on how reasonable that rotamer is, right? But those rotamers are based on the assumption that you have the L stereochemistry. So if you have the D stereochemistry, what do you think is going to happen to all those scores, or all those Ramachandran plots, really?
Incorrect.
Right, they're incorrect, yeah. But it's actually kind of simple. If you since this is just a mirror image, you can just kind of flip those, the data around. And so, if the rotamer, your best rotamer, say, was at like 170 degrees, you can now swing it around the other direction to 170 degrees. I mean, negative 170 degrees, and you have the same results. So that's all they do. For Rosetta, when Rosetta sees a D-amino acid, it has a little note in the file that says, I'm a D-amino acid, use the inverse of the Dumbrack libraries to score this amino acid. So that's a really quick fix for that little problem case. Okay, so Dr. Gray mentioned Bonneau's lab and how they did these things called peptoids. And what peptoids are, they look like this. Right. Very similar structure, although I didn't flip it upside down. So what's the difference here between these two molecules? Right. Side chain is no longer on the C alpha, it's actually on the nitrogen. So they made peptoids for every one of the 20 canonical amino acids, but the R group is moved from here to here. So in this case, Rosetta can use all the same methods of handling phi, psi, and omega, because phi, psi, and omega are still here. But now our R group's different. So there's a couple major differences that you have to consider when you're trying to model one of these as opposed to a regular peptide. Like before, your rotamers are going to be completely wrong because they're not in the same place as they were before. So, but in this case, you can't just take the mirror image. You can't just flip it. So it's really important to generate a new rotamer library. Now, we have a PDB that's over 100,000 structures right now, so there's a lot of data to mine to get information about rotamers. For peptoids, there's a very small number of peptoids that have been crystallized. So we can't rely on the statistical data. We actually have to model the stuff using quantum mechanics. So what they usually do is they'll plot. So down here you have your angle, actually torsion angle, and then you have your energy. You get some sort of plots like this, and you visually pick your minima, and you plug that in and say, okay, this is one rotamer, that's another rotamer, and that's kind of what you put into the database. So you actually have to go through every peptoid and generate these manually. Yes?
Has anyone tried doing that for the regular peptides and comparing to the Dumbrack library?
I believe so, yes. Right.
So which is better?
I'm not sure how exactly they compare, but I'm almost certain they've done that comparison.
Yeah. Oh, yeah, that's a good question. You're asking, does it match what proteins really do? The statistics from the PDB are that. So that's kind of your gold standard.
Mm-hmm.
If you're asking, does it capture the energy of the side chain itself, the quantum might be better at that, and then the statistics are capturing also nearest neighbor interactions and the polymer presence and everything else. They're pretty close, though. Brian Coolman's worked on this, and so is Rich Bonnell. Both of them. Actually, it was the same student. Got his PhD with Brian Coolman, and then went up to Rich Bonnell's lab. So there's a paper about this where they've compared the quantum with the rotamers.
So they did that to generate rotamer libraries for the R groups that are on the peptoids. There's another interesting thing that comes into place here. So, here again, we have phi, psi, and omega. So, do we ever do much with modeling of omega in Rosetta? And why is that? What's that?
Yeah, so omega is pretty much constant.
Yeah, for the most part, it has like two possibilities really, right? Because you have partial double bond character here, so there's a high energy barrier for flipping it. And if I were to extend this, if I were to extend this guy a little further, you can see that you want this to be like a trans relationship across this partial double bond, and that keeps this part from bumping into this part. So for steric reasons, your 180-degree omega angle is usually favored, so we generally don't model that. You can turn on the option to model it in Rosetta, and there are cases, like especially with proline, where you sometimes get the cis form instead of the trans form, but for the most part, that doesn't move around much. But look at how it's different over here. So here's my omega from the earlier residue. I have an R group here, and I have an R group down here. So who's to say whether cis or trans is better in this case, or even what cis and trans are actually kind of meaningless in this case. So who's to say whether 0 degrees or 180 degrees are better in this case? So since you don't know, you have to model omega. So you have to turn on specifically the modeling and sampling for small movers and such of omega when you're dealing with peptoids if you want to get answers that are going to match reality. So that's just another thing to keep in mind. Unfortunately, in Rosetta, so much of the code has been written assuming peptides that a lot of times when you have a new system like this, you have to go and rewrite the code to allow it to pay attention to omega. And that's kind of a problem that I run into a lot with the sugars, is a lot of assumptions that I had to kind of get rid of. Okay, here's another interesting one. All right, these are called beta-peptides. So that looks a lot like an N. It's supposed to be a screw. Okay, what's different here? Right. We've added an extra carbon. So we have our carbonyl, and then our C alpha and our C beta. And it's called the beta peptide because the nitrogen is on the C beta as opposed to C alpha. The R group is on the C beta. You can also put the R groups in the C alpha. That's another kind of beta peptide. But all right. So here, what they did, I don't know if this was not necessarily the best thing, but this is what they started with. They used the exact same rotamer libraries they did for regular amino acids. They figured, at least from this part of the molecule, it's similar enough that they're going to try to stick with the same rotamer libraries. They noted in their papers that we could improve on that. We could generate a new rotamer library, but for now we're just going to use the regular amino acid ones. The biggest issue is what you do with this extra atom here, because now we have, well, I don't even know what they call it, but you have phi, psi, and omega, so you have this new torsion angle right here that you now have to deal with. So if you have any algorithms in Rosetta that assume that you only have 3 backbone torsions, you now have a major problem because you've got 4 to deal with in this case. And that's going to be the problem with a lot of these alternative non-amino acids. Is, you're not limited to just 3 backbone torsions that you have to sample when you do your small moves and your shear moves. So that's something that had to be you have to keep in mind. Okay, going back over here. This one is not going to look like, you're not going to see the amino acid similarity at first. You look at it a little carefully. Call it R1, call it R2. Right. Okay, this is 2 residues. This one is just, is entirely a regular amino acid, except there's something off this nitrogen. And this one is the same thing. Here it's a regular amino acid and C-alpha carbonyl, but you got these 2 methylene groups that are branching to form a 6-membered ring. So these are called oxopiperazines, and because they're usually oligomers, they call them OOPs for oligo-oxopiperazines. It's kind of a fun acronym. So this is an OOPs molecule. This poses some additional problems, right? So your R groups you can use the same rotamer libraries because it's just the same. You have the same amount of backbone torsions. So what problem do you think you might run into, though, if you started changing phi and psi in this case? So if I'm on again, if I go and do a small move and I change my psi angle in this case, what's going to happen to the molecule? Yeah, you're going to rip your ring apart, right? Because now you're looped up. Unfortunately, so Rosetta was built with this idea of this full tree with branches and didn't really take any consideration into rings. So you can't change these torsions without also changing the ring torsion to repair it. So what they had to do is they had to put a line of code in that if the residue you're dealing with is an oops, and you modify phi or psi, you then have to run a special mover that goes and corrects the broken ring and has to put those atoms back where they need to go and twist the hydrogens around and do a minimization. So you have to take care whenever you have rings in your residues to make sure, because just changing these torsion angles is not enough. That's another major concern. So basically every one of those concerns I just talked about is present in carbohydrates. Yes?
So are these, so what are the big cases where these are seen or important?
Yeah, that's a very good question. I'm glad you brought that up. So I started out just talking about things that are in biology. All these examples are not in biology, except for a few exceptions. Right, yeah. What's that?
Okay.
Yeah, except for a few exceptions, or in usually like in antibiotics, things aren't made by ribosomes; they're made by non-peptide synthetases. So they're oligopeptides, but not polypeptides. So these are made though as mimics for peptides. So you can make. There's another example I didn't show, but this is actually a sort of good one. If you put this here, you add some new structure to your molecule. So if you have an unstable peptide, you could then synthetically add this kind of a portion to it. And now you have a more stable section of the molecule that will fold and maintain the fold you want to have. Some of it is just sort of basic science, sort of what happens if we made a protein that was all D instead of L, what would happen? Would it do exactly the opposite chemistry? And that turns out to be the case. If you make an entirely D protein, it will do the opposite stereochemistry enzyme reactions. And sometimes people make mimics for RNA and DNA as well, and what the idea is those structures will go in and bind to DNA exactly the same way the RNA or DNA would, and you can directly influence or deliver a drug to a part of DNA, to intercalate. There's all sorts of different possibilities. A lot of this is bioengineering kind of stuff. But we want to be able to do both in Rosetta, model the natural as well as the engineered.
There's advantage to these, right? If you just put a peptide in a cell, it's going to get
yes.
by proteases and chopped up. A protease isn't going to be able to chop this up because the chemistry is different. It doesn't bind the same. So you can use these as drugs to bind to things like the way peptides bind, so the peptidomimetics, but the body doesn't metabolize in the same way, so it changes how you can deliver them and how they persist in the body.
Yeah, and there's even people who are also trying to make synthetic life, like kind of make cells with all these artificial kinds of things and see if they can kind of like make a completely man-made cell using all different stuff. Just again, basic science kind of how can stuff work problem. There's a lot you can do. All right, so the next example though is entirely biological, but people mess around with these as well. So here is a typical oligosaccharide. Okay, this is just, we'll make this one go. This 1, 2, 3, 4. Yeah, out here. Okay, so this is 2 residues. So here's where the residue would break. That's residue 1, this is residue 2. And these are the stereochemistry could vary at all these different positions, give you various glucoses, galactose, mannose, whatever. But this is your typical sugar system you see in most biological cases. You can also get 5-membered rings and other crazy stuff as well, but we'll just start here for now. Okay, so what is different? There's a billion answers. So what's different in this case than the case of, say, a peptide residue type? What sort of differences do we have here? In a peptide case, we've asked questions about the main chain. We've asked questions about our rotamers. So what is the main chain in a case like this? Yeah?
Well, because it's a ring, there's really a straight chain
Right.
necessarily. It
Right.
goes both directions on there.
Yep. So in this case, the main chain is sort of ambiguous or arbitrary. We can decide what we want the main chain to be. Just like over here, the way it's treated in Rosetta is that this is treated as the main chain, and these are treated as 2 additional atoms that we have to worry about making sure are closed. And I do the same thing in the case of sugars. So I actually put a cut here, and I treat this as the main chain, and I put another cut there. Well, actually, that's not where it was. Sorry. Here. The reason I picked that is not entirely arbitrary. I did this because biosynthetically, when sugars cyclize, this is the position where they cyclize. So they start out as a linear hexose, a 6-carbon sugar, and it cyclizes in a 6-membered ring by connecting oxygen number 5 to carbon number 1. That's how the chemistry happens. So that's where I chose to put it to kind of mimic biology. But we could have gone, as far as Rosetta is concerned, we could have drawn the main chain through a different way. So the main chain goes through like this. Okay, so that's one interesting thing. What about rotamers? What's immediately different from the case of the rotamers I would need to have for this molecule and this molecule over here? Yeah?
Well, these would be like conformations in the rotamers, like I guess like chair and boat and all the different types of conformations that a ring can take.
Yeah, that's one, almost like a completely separate thing from rotamers, really, in the way I think of it. But yeah, ring conformations are important. In addition to your, because here you have, there's different kinds of main chain backbone torsions right now. So I've got this torsion, this torsion, this torsion. When I get to this torsion, this one is just kind of like freely spinning like a typical backbone torsion. And in fact, they actually do call these phi and psi. So this is psi and this is phi. But twisting these doesn't affect the ring, but twisting these ones does affect the ring, just like they did over here. So all of the ring torsions have to happen kind of simultaneously. If I change psi, I get a new molecule, and that's just totally fine. I didn't violate any chemical rules. If I twist this, which is called nu 1, that's actually going to rip my ring apart. And in fact, geometrically, if you change nu 1, in order to maintain the ring, you also have to change nu 3 and nu 5 to make sure you maintain your ring in a proper form. And so what I've actually done to sort of solve this problem is I have a ring conformer mover. And what it does, it goes to a database of all the possible mathematically ideal ring forms. So I don't know if you remember from organic chemistry, these chairs and boat forms. Well, there's 2 of these, and those are the minima. There's also the boats, which are some of your maxima. But there's also twist boats, which to draw on, I should have brought a model actually, that's what I should have done. There's like 15 different ones. There's twist boats, there's envelopes. Envelopes kind of look, that's a 5-membered ring, so an envelope kind of looks like that. There's all sorts of different conformations. And in this case, all the torsion angles are the same all the way around the ring, and here, like, some of them are 60 degrees and some of them are 30 degrees. Here, so you got some are 60, 15, and 0. So like, there's a pattern, and there's 15 mathematical combinations you can get for a 6-membered ring of ideal conformations. And so what I do, rather than sampling kind of like how we sample the rotamer libraries, I sample the ring conformer library, and I pick one of those 15 idealized ring conformations and set all the internal angles in the ring, the torsion angles, and then I minimize, and that's how you make a move of a ring for a sugar. So it's a completely new kind of move that is unique to any kind of ring molecule in Rosetta. Now, another question for you is, if you can kind of tell by looking at these pictures, I really should have brought a model. If we know this is stable because all of these positions, and if you remember from organic chemistry, all these positions maintain their tetrahedral quality, so that the bond angle is still 109.5 degrees at all these positions, right? And in the ring, so even in here, it's really close to 109.5 degrees. If I have these 5 atoms all planar like this in this envelope, so I have, essentially, this half of the molecule is like a hexagon over here. What angle do you want to have right there?
120.
Right, 120. But a carbon wants to be 109.5 degrees. So if I change all my new torsion angles, which are again the angles like this, if I am twisting all these to make this ideal ring conformation, all of a sudden, Rosetta, it still has this bond here set at 109, even though it needs to be 120 to actually be flat. So usually in Rosetta, we only change torsion angles, but when you change a ring in Rosetta, you have to also change the bond angles, or you get completely ridiculous structures. So when I do this ring mover, it changes all the necessary torsion angles and then changes all the necessary bond angles, and only then do you actually have an ideal structure that makes sense. So it's just a new consideration that you have to deal with, that things become a lot more complicated when you add rings into the picture for any molecule like this, any residue. Okay, so that's two kinds of sampling. We've talked about backbone sampling, and we've talked about the ring sampling, but you also have these other rotamers. So here we have a chi, and this would be another chi. That's chi 2, and this is chi 3. You have chis in amino acids as well, but we have a single side chain in the case of an amino acid, right? We have just this one R group. And if there's multiple chis, they're just further on that same branch. But each of these sugars has, this is a chi 2, chi 3, and then I've got up here, this is chi 5, and then I have chi 6 up there. So I have, not only do I have multiple chis, but I have multiple side chains. So this becomes really crazy because this one can turn around independently from this one over here. But if these two are twisting around, they might have, this one might actually have a dependence on where this one is. Because you could form, if you had a hydrogen pointing out like this, a sort of intramolecular hydrogen bonding kind of 5-membered ring thing going on here. So it's important. So they're not independent of each other. Some of them aren't independent, but some of them are. So the rotamer libraries get really complicated and confusing. I haven't yet solved this problem. It's just something I'm very aware of being there. Right now, I've exhaustively kind of sample every 120 degrees and kind of just score them to see which one's the best. At some point, I plan to try to make a rotamer type library for the sugars. It's just another consideration, though. We got really lucky when we started with amino acids. In fact, there's only one side chain, but a lot of molecules have more. Another example would be RNA or DNA, right? There's all sorts of different side chains there as well. So, let's see.
So there's no sort of like optimum hydrogen location that you would use?
Yeah. And there are far fewer structures in the database for sugars than there are peptides. But there are some, and people have started going through these and tabulating sort of information like what is the most stable form for this particular residue or this one. And I'm starting to go through and add that into the Rosetta database. That's what I'm working on. But there's a lot more work to be done. But I'm just right now just kind of painting the kinds of problems that you have to deal with when you're thinking about new molecules to add in. One of 3 problems. And we've been focusing mostly on sampling issues. How can you make sure as you cover all the degrees of freedom? So, an amino acid has the phi and the psi, those are 2 degrees of freedom, and all the chi angles, we have those degrees of freedom. The sugar has all the different ring degrees of freedom, it has backbones, it's got all the extra side chains, there's way more degrees of freedom. For that reason, the sugar is more floppy, it usually doesn't resolve as well in a crystal structure. So it can be more challenging to make sure you correctly sample all the possibilities that molecule can make. But we also have scoring. It's important to make sure that we consider these sort of problems as well. In the case of the peptoids and the beta peptides, most of the scoring is pretty simple because we're using the same general kinds of atoms, right? There's not really new chemistry going on. In the case of the sugars, we have a lot of new kinds of chemistry going on. Like, how do you handle that carbon right there? It's got an oxygen here and an oxygen here, so you have a lot of electronegativity being at these 2 positions, pulling the electrons away from that center. So that particular carbon is very delta positive. So that's going to change the way it binds to things and how it affects the local chemistry. We have a lot of OH groups, so we can use the same kind of OH kind of chemistry that we get from serine. That's not a problem. But like I said, this one's kind of tricky. Like, what do you do with that? And what do you do with the hydrogen that's sticking out right here compared to the one that's sticking out over here? And they actually have some interesting effects on each other because of the fact they're pointing at each other from across the ring. And there's a lot of really weird effects in sugars called like the anomeric effect that affect how the hydrogens behave. And in some cases, some people argue for crazy things like hydrogen bonds from a carbon to an oxygen as opposed to. So, talk about things like this. Some scientists are arguing that that is actually a legit thing that happens in carbohydrate chemistry because there's actually enough of a difference in polarity between these two atoms. So there's a lot of stuff to consider that needs to be taken into account. So for the people who worked in the peptoid lab and Bonneau lab, in addition to the peptoids and such, they also deal with non-canonical amino acids and post-translational modifications. And so for example, what do you do if you have okay, so this is parahydroxyphenylglycine. So it looks at first like tyrosine, but you're actually missing a carbon here. This is, again, a natural amino acid. It does occur in biology. It's in a lot of antibiotic drugs. So it is floating around in our cells, or in some kinds of cells. But what do you do with something like this? Again, you have to generate your own rotamer libraries, but you also have to deal with, you might have to deal with special chemistry. There's also ones like this. Or there are molecules, I think they have in there, that look like this with chlorines. The regular amino acids don't have chlorine attached. What do you do with these atoms? So you have to make sure that you have the right, van der Waals information for the chlorines, and all your potentials have to be correct. So what they actually did is they went through the CHARM database and took all the CHARM parameters, and they entered those into Rosetta. So that for things like chlorines that are on your special non-canonical amino acids, you can have that information. The parameter file tells Rosetta how to use it and which terms to use. And you can use what's called an MM standard score function as an alternative to Polaris 2013. And it will take into account the torsion angles and the bond lengths of all these atoms by looking through the database and say, hey, every time you see a chlorine next to a carbon, next to another carbon, then use this sort of score for that bond that you measure. So that's kind of what they do in that lab to deal with special scoring problems. The other issue comes with the scoring methods. We already hinted on some of this, like the Dunbrack scoring library gives you a score in Rosetta based on which rotamer you're using. So if you're dealing with something that doesn't apply, you're going to just get a 0 in that part of the scoring function. And so in my case, I have phi and psi. I can't just use the typical Ramachandran plot because it totally doesn't apply to sugars. So for the Ramachandran score, I have to kind of have an entirely different score that I get for sugars. And in fact, there's a scientist in Wood's lab who's done that. He's gone through all of the structures of the carbohydrates and plotted sort of like Ramachandran plots for them and extracted that information out and has a sort of a phi-psi probability kind of score that can be applied to carbohydrates. So I'm going to be implementing that in Rosetta pretty soon. There's one other sampling issue I completely forgot about that is kind of unique to sugars. In a polypeptide, you have a single linear polymer, right? So you start at your N, you go to your C, just a linear molecule. In the case of sugars, everywhere I have all these side chain OHs, there could be another sugar branching off. So I could have another sugar here, and I could have another sugar out in this direction, which means I have this crazy branching thing going on. The full tree, at least all the full trees you've seen in class so far, are all linear. Well, you can actually make a branch full tree in Rosetta. I don't think we're going to be we're not going to be doing that today. But you can make branch full trees in Rosetta. But it becomes a little tricky because all of a sudden what used to be a side chain is now actually considered a main chain of a second branch. So the way I deal with that in Rosetta is with patch files, which I'll talk about in just a second. Go ahead.
Is the branched full tree still directed?
Yes. It can be cyclic, right? So it's still directed. So you start, it's kind of like, again, a tree. So you start here and you just keep going off in different directions, but it's all
You won't have more than 2 branches coming together. Or something.
right. Right. Yep.
So,
Yeah.
so related to that, especially for like glycosylation, you don't always have the same sugars.
Mm-hmm.
So stochastic, whether you have like distribution of sugars, right?
Right.
You
Well.
probably that as well.
Yeah. So that's exactly correct. In biology, it's not. There's no like DNA. There's no central dogma for sugars, right? You don't like have every protein doesn't have the exact same sugar glycan patterns all around it. They tend to be they're pattern and trends, but a lot of that's based on biosynthetic flux as opposed to DNA. But obviously, when we're modeling something, we're modeling a single molecule. So what you could do is you just kind of have to pick one to model, and you can maybe model a bunch of other ones and compare them all, see which have the best energy, depending on what your application is. The other thing is that they're so floppy, and there's so many degrees of freedom that one structure is probably not like the correct answer. So what we are going to be doing is generating more ensembles of decoys. So here's kind of this wide area, like a cloud, where that glycan can sweep out. And hope maybe we can learn something from that ensemble as opposed to saying this is the correct structure. That's the sort of thing that we would be doing. All right, so there's scoring, there's sampling, and then there's the database size. So there's 20 canonical amino acids, not so big. There's 4 nucleic acid, RNA nucleic acids, and 4 for DNA as well. So we're talking about really small database sets for your starting residue types. But what about with sugars for a 6-membered ring? I've got all these stereocenters. There's like 16 different possible 6-membered sugar rings. And then depending on the branching that happens, like if the main chain is going this way, or the main chain is going this way, or over here, I have 4 different possible main chains I could build off that sugar. For Rosetta needs to know the direction of the main chain, so it has to be a different residue type for every one of those sugars. So all of a sudden, I've got this huge, huge amount of sugars I need to keep in the database. Now there is a shortcut. Have you learned about patch files at all yet in Rosetta? Are you aware of them? Okay. Well, we're going to cover those in our workshop, so I'm mostly just going to stop. We're going to move on to the next thing. But a patch file is basically a way in Rosetta of taking a base residue type, like an alanine, or say a serine, okay? And let's say you want to make a phosphoserine, which is just like serine, but has a phosphate group on there. Since everything else about the phosphoserine is the same as regular serine, what Rosetta does, it just uses a serine, and then it takes a patch file and it applies that patch to the serine. And the patch file is simply just a list of instructions on how to turn that serine into a phosphoserine. So if I had a serine here, the patch file, keep doing that. The patch file will say, first delete the hydrogen, add a phosphorus, change the oxygen to phosphorus bond length, add another couple oxygens with double bonds, and some charges. It's basically step by step. It says delete this atom, add this atom, change this bond length, add this bond angle, and it builds the rest of the molecule from the serine. And that's how patches work. And in fact, you may have noticed, has anyone noticed when you print a pose out, how residue 1 and residue and the last residue always have an additional little bunch of text added to them? That is basically the Rosetta's way of telling you this is a patched residue. So if you have an alanine at residue 1, you don't really have an alanine residue. You have a N-terminal alanine residue, which is a totally different residue type because you have new atoms. You've taken the lower connect, deleted the lower connect, and you've added a hydrogen. So and you've also then changed the bond angles of those hydrogens. So that's what a patch file does.